to introduce myself. So my name is Val Arboleda. I am an assistant professor here at UCLA. I'm in the departments of pathology and lab medicine. I'm a physician scientist, so I do a little bit of like actual clinical pathology, but I'm also in the departments of human genetics and computational medicine. And so I know I'm the last thing between all of you and the beach. And so I was going to kind of give a fairly light presentation, and I think it actually jives pretty well with some of the ones that we've heard up until today. One of the things that I think about a lot, and I'm sort of, when I was thinking about what to present today and sort of what to share with you all about the work we do, is sort of thinking about the lens that I take as a pathologist. So from the clinical perspective, I spent probably the first parts of my career really thinking about diagnostics and patients as new sequencing technologies was emerging. And so most people, when I tell them I'm a pathologist, their eyes sort of glaze over and they're like, I think I know what that is, but I'm not 100% sure. So I'll kind of spell it out. We are these very intellectual looking people looking at microscopes and looking at tissues. We're very thoughtful. We like to think hard about disease. But we're trained as physicians. We do a full residency, but we don't see any patients. We will do autopsies. So if you watch any of those like criminal investigation shows, that's pretty much, those are pathologists who are doing autopsies and figuring out how and why you died. We look at tissues and cells under the microscope to identify cancer and other diseases. So that's most commonly where people interface with pathology. But generally, you don't see pathologists because we're usually in our office, and our office is in the basement. There's no windows. We don't see patients. And so that's our general ethos. These offices, as you can see, there's maybe one or two of them have windows. The other things pathologists do, and this is something that this sort of more on my expertise side, is we run clinical laboratories. So anytime you go and get a blood draw, that blood goes into sort of this black box, and you come out with a result. And in that black box space, things happen to your blood sample so that you get a result. And it's a very regulated space. It's a space where any clinical discoveries or any sort of research things that you think about and you're like, oh, this would be great for diagnostic, when your blood or any sample, a urine sample, what have you, goes into the pathology lab, we take that sample, we take that really exciting test, and we drill it down to its core components, and we spit out a result. Like for your COVID test, if you give us a nose swab, we'll tell you something like positive or negative. There's a ton of information that we get when we do that diagnosis that don't actually get shared with the patient in the effort of making things simple and straightforward and sort of uniform across things. So I like to think about it, so I'm very much a researcher. I love the discovery phase. And so I think about it a lot like, well, pathologists, we take these really exciting tools that everyone's been describing and that all of you work on, and we make them usable to anyone and everyone. The physician who graduated from medical school 50 years ago when sequencing didn't even exist, we make the tools accessible and reportable so that when your doctor has 15 minutes with you to give you a result, you can digest it and they can act on it, right? So that's kind of where pathology sits in the world of medicine. And I think a lot about how some of the tools that we're creating here from the computational tools will really translate back into the clinic and what's required in that space. And so my research lab is focused around rare genetic disorders, and that's sort of where my career started. As a graduate student, I studied a lot of really rare congenital disorders. And my lab right now is sort of split. We do about half of our lab is genetic disease modeling, and then the other half of my lab is really interested in sort of the architecture of disease. And so today I'm going to share with you I think some thoughts around clinical phenotyping in whole genome sequencing cohort and in biobanks, because I think there's a lot of room for improvement around methods that are tailored and geared towards patients who harbor rare genetic conditions. And I'll sort of try to give you a flavor of what's possible, because often people will think of germline disorders and pediatric disorders as the low-hanging fruit. We actually have tests in the clinic. We can provide a genetic diagnosis for a large fraction, but there's still so much more that I think we don't understand, and we can use the data the really rich and sort of very manual data curation from rare disease patients to better understand how genetics express and change phenotypes. So I'm going to start today with a background so we're all on the same page, because I know everyone here comes from either very mathy backgrounds or computational and sort of different areas within computational genomics. So we'll talk a little bit about a definition of rare monogenic disorders, and then we'll talk about sort of one case study of where we can do this integration of common variants and rare variants around Mendelian traits. And so this is one case example. There's a couple more that I'm happy to kind of talk about more, but I really wanted to keep the focus around phenotypes and Mendelian disorders. So we're all here for genomics, and I think what we know is that sequencing is easy. There's these machines that Illumina makes that can see these NovaSeqs, 20 billion reads over 24 hours, and you can sequence pretty much anything. You can make a library out of anything, garbage in, garbage out, but regardless, you can still sequence it. You can even go out into the field with your iPhone. I've never tried these smidge ions, but I always really like the fact that they can be plugged into what looks now like a very old iPhone. But interpretation is really hard. This is something we grapple with even with the most straightforward cases in the clinical lab. This is what we sort of, at the end of sort of the most straightforward pipelines, you get these VCF files that have hundreds of thousands of variants and all this information, and sort of, if you were to try and open it, well, don't open it as an Excel file, but if you were to try to do that, your eyes would probably look like this. And that's sort of what we grapple with when you take this translation from the research space into the clinical lab, is you have to take this information and distill it down to something that's graphically usable for the average person who's not a computational biologist, not a genomicist, not somebody who's like running code through a pipeline. The vast majority of pathologists, I would say, probably spend most of their time doing medical training, and most of us are not sitting there on our Linux computers trying to go through this data set and parse it. So some definitions just to start us off. Mendelian disorders are single gene disorders. They're often referred to as monogenic disorders, and we define them with some of these traits. They're rare in the population. So we don't see a lot of people. There's a formal definition, which I'll talk about, for what the FDA defines as a rare condition. But for a lot of the really rare genetic rare conditions, we're talking somewhere around a couple thousand people in the United States maybe are diagnosed. These are often single gene mutations, and sort of the biological definition is that mutation is both necessary and sufficient to cause whatever disease you're referring to, disease or phenotype. Often the mutations that we're looking at and the genes we're looking at are protein coding. I would say like 99.95% of the time, these are protein coding and variants, and they have a really high effect. And that's meaning that they have a high penetrance. If you have these mutations and it's autosomal dominant, you will have this disease. I mean, how do we find them? Generally, we find them. So in the past, like when I was a grad student, not that long ago, but now it's like been 15 years, we did linkage and we would sequence our favorite peak. And those peaks would maybe be 20 megabases, and that was like a huge deal. Nowadays, we don't even bother with linkage. We just do exome sequencing and do some IBD calling around STRs. But exome sequencing is generally in families is the way that a lot of these are discovered. The FDA has a formal definition for what they call a rare disease, and this has to do with sort of some credits around how easy it is to get FDA approval for these orphan diseases is what they're called. And so this is a disease or condition that affects less than 200,000 people in the United States. And that's actually, it's depending on how you look at it, that's a lot of people, 200,000 people with any given rare genetic disorder. That's way more than most genetic diseases for one disorder. So it actually starts to cross into the world of some of the rare complex traits, like some of the autoimmune disorders. But to put that into numbers, it's about like 1 in 1,600 people. If there were actually 200,000 people in the United States, there's 330 people in the US. It's about 1 in 1,600 people will have that particular disorder, or 0.06% of the population. But sort of that said, there's a lot of different rare diseases, and if you sort of add everyone together who has any rare disease, it's estimated that it's around 10% of the population, and that sort of takes in some gross assumptions. So when you start to think about it, well, there must be people who have these rare genetic conditions combined with some complex traits or some variants that are going to influence how that particular rare disease comes about. And so over the last 10 years, we've really come a long way with clinical exome sequencing. It used to be this sort of niche kind of test, but now it's offered pretty routinely at academic labs, but also in industry like Ambry, Invitae. There's a number of companies that will offer these tests, whole genome, whole exome, or some small targeted panels, depending on essentially what your insurance will cover. And with all of this sort of the diagnostic yield for exome, this is sort of the glass half full approach. The diagnostic yield is roughly 25 to 40%, depending on which paper you're looking at, right? And so in the medical genetics world, people who were seeing patients were like, this is awesome. We are actually diagnosing patients with their genetic condition. We're able to lump all these kids who we didn't really know what they had. We knew they just were developmentally delayed. And we now know these all have this condition X and condition Y based on the genome. But sort of, I look at this and I'm like, I'm still sort of puzzled because germline disorders were always meant to be sort of the low-hanging fruit. This is the stuff that should be easy. There should be a single variant that you can find. If it's a really rare, severe condition, ostensibly the parents are unaffected, and so you can use the parents as controls, and that you know that this new mutation is new in the germline. It's a new mutation that arose in that kid. And so the glass half empty approach is like, well, the diagnosis failed 60 to 75% of the time, and why is it continually failing? We have all these tools, we're sequencing all of the known protein coding genes. So what's still missing? We can find these, but these cases are rare. The cases are rare, they're spread all over the world, and so it really takes a little bit of serendipity to identify these variants and sort of some of the challenges around how we phenotype them. Because when clinicians are seeing patients, they're thinking, okay, I have this, I found this gene, does it match with the people who have been reported beforehand? So a lot of this, there's a little bit of sampling bias, I would say, in how we define these disorders. So one of the things that happened when I was a resident here is that we identified, sort of serendipitously, we were in these genome data boards where we discuss all the cases, and over the course of maybe 3 to 4 months, we identified, we saw this one gene come up several times as a de novo mutation in the gene CAT6A. And so we found there were 3 cases in a row over the course of 6 months, and we don't review that many cases. So if you have a good memory, you kind of remember these genes. You're like, that sounds familiar. I swear we saw that last week. And it turns out that there were all these mutations. 3 of them happened to be at the exact same position within that gene. They were all what we call truncating mutations. So they change an amino acid into a stop codon. And they all are de novo mutations. And what that means is that the parents up here, can you see my, well, I can't even see where okay. So the parents up here, 1 and 2, are unaffected, and then here the proband is affected. And all of the parents did not carry this mutation. All of the kids had severe developmental delay. Some of them had cardiac defects. And then 3 of the 4 patients that were identified harbor the exact same mutation. And so what we realized when we started this process was I contacted all the families, and we kind of asked the question, well, what other phenotypes do your kids have? Let's get a good phenotype for all the other kids who will be diagnosed after this so we can do a good comparison. But there's a real challenge when you do the first sets of cases, right? The sampling bias that I was referring to is that when you define a disease by the first 3 cases that someone happened to see, you're often defining sort of the most severe clinical cases. And you sort of make the assumption that anyone who has this particular mutation in this gene, this type of mutation in this gene, must be sort of devastatingly affected. And over time, what you sort of realize is the phenotypes we see, people keep adding more and more phenotypes on. And we never can quite tell if the phenotypes are truly associated with this one gene as you get to like your couple hundred patients in, or if these kids are seeing the doctor so many times, they're just getting extra things added onto their clinical phenotypes because they see a doctor every other week, and someone's going to annotate something and note that there's an extra issue with the eye that has not been seen. And so this issue of differential phenotyping, I think, is a really important one to think about, especially as we're going through and doing these genetic assays. The other thing that I want to kind of touch upon now, and we'll talk a little bit more about later, are thinking about your control group. So when we think about the mutations, we often use as the control the parents, because the parents are unaffected, they're related, and if you see the same mutation in the parents and the parents don't have an effect, we assume that because these are very highly penetrant disorders, that whatever mutation you're looking at must not be the causal one. But we don't define normal well. So if in an ideal world, what you'd want to do is if I found a kid and I said, well, this kid has a congenital heart defect, and I wanted to know what a good control would be, I would do the same assays that I did on the child, looking at the structure of the heart with CT scans. I would do the same thing on any given control. But we often don't do that. We kind of just say the absence of needing any clinical treatment is enough to say that you're quote unquote normal, which is good, right? It's a binary between clinical disease and not disease, but it doesn't mean that there are no milder structural anomalies that are sort of subclinical and below detection within a person or a cohort, and it can sort of alter the way we think about penetrance and expressivity in our assays. So I should have defined penetrance for you. Penetrance is something we talk about a lot in monogenic disorders and in human genetics, in medical genetics clinic. And so it's for those who have a pathogenic proportion, what proportion of those individuals have manifestations of disease. So if you have a whole family tree and every single person in that family tree inherited this one mutation, how many actually have the phenotype that you're interested in? It could be a disease, it could be hair color, whatever it is that you're looking at, that's penetrance. And for Mendelian disorders and anything that we generally diagnose in clinic, people don't like non-fully penetrant disorders because it sort of imbues a level of uncertainty that clinicians are often very uncomfortable with. So we really in clinical genetics, we actually will refuse cases if it doesn't seem like the disorder is monogenic or fully penetrant, because it's a lot harder to identify. Your list of genes will be much longer if you're saying, oh, it may not be penetrant, maybe this is the right one, and you'll have a lot longer of a list, and you give it back to a patient and they can say, oh, well, I don't know which one it is, and it leads to a lot of confusion and a lot of anxiety around for the families. The other thing we think a lot about is expressivity. So if within that same large pedigree, everybody who has a mutation, some people will express the disease more severely than others. So let's take the example of a monogenic disorder that affects your cholesterol. Some will actually have very high cholesterol levels and some will have lower cholesterol levels, and it's really unclear. Sometimes it's unclear exactly why that happens. Maybe some of it is due to environment, but there's a lot of challenges around this. So for common disorders, penetrance and expressivity, you can collect enough samples that this is fairly straightforward. UK Biobank is a great example, and it's one of the resources we've been using a lot to think about penetrance and expressivity. But in rare diseases, there's a real logistical challenge, right? If there's only 300 patients in the world, how do you study them? So patients who have mutations in CAT6A, there's roughly 330 individuals around the world that we know of based on the foundation that sort of collects and collates this information. And it's challenging to think about how you might study these disorders from that perspective. And so we've been thinking a lot about both phenotyping and using endophenotypes and sort of trying to partition out the phenotypes in a way that would make sense. So we already talked about this, sorry. But when I was sort of discussing about CAT6A, we have this sort of early phenotype, the most severe cases, but then 3 or 4 years later, we went ahead and we redid our collection, and we identified 72 cases, 20-some who had been already reported in the literature. And what you can see here is that on the bottom, there are these cases in which, I'm not even going to use my slide. So here on the bottom there are these cases we've already reported, and they're all sort of towards the end of the gene. And these tend to be pretty severe cases. But as we went through and we actually paired up with the UK Deciphering Developmental Disorders cohort, they collated, they sequenced about 10,000 kids with various forms of developmental delay. And then we asked them for all the kids with Cat6A, and we phenotyped all those kids. And it turns out they found a whole lot more mutations all across the gene. Most of them are still the truncating mutations. Most of them are still de novo, but we had a couple that were inherited. So there were a couple individuals in whom the father had a mutation, and then all 3 kids had the mutation. We identified more missense mutations. And what came with this sort of expanded genotypic spectrum, we said, well, they probably are all the same. But the family started chatting online, and they said, you know, we seem pretty different. Like some of the kids are walking and talking and like going to school and, you know, playing baseball. They're delayed, but not to the level of the first kids that we saw. The first kids we saw with this condition, I think to this day, still have never spoken their first word or walked or toilet trained. And so that sort of range of phenotypes was sort of a little bit telling to us. And on the side of here, there's another, this gene has a family member called Cat6B, and it actually gives rise to 2 syndromes that are unique. One is called genitopatellar syndrome, and the other one is called, it has this horrible name, Smith-Biesecker-Barber, I think I'm forgetting one name, Say of Odo syndrome. So it has this SBBYS. And I forgot Young. And it gives rise to 2 independent syndromes that actually were clinically identified first as different, and then when we started sequencing patients, they realized that both syndromes were due to truncating mutations in the Cat6B gene. So as we started to look more, when patients started telling us that they thought that there were some differences in phenotypes, we started to look more closely at what we consider late and early truncating mutations. So there's some, we're not going to talk, this is more of like a molecular mechanism thing, but early truncating in the mutations in a gene are thought to lead to nonsense-mediated decay. So that the cell realizes that there's a truncating mutation, it's the wrong copy, and they should just chew it up so that it never gets actually expressed as a protein. But mutations that are in the last exon of a gene are thought to escape this mechanism, and so that sort of truncated version of the protein will be expressed and can actually have these aberrant effects. And it's thought that maybe that this sort of shortened version of the protein has either dominant negative or gain of function effects. And that might give rise to these differences in phenotypes. So when we looked at the individuals who are here with the expansion of phenotypes, we have a column for early, a column for late, and then we ask in what percentage of those individuals is that phenotype present. And you can see generally the later truncating mutations are more likely to have any of the given phenotypes that we listed here. The other thing we noted was that there were a lot of one-offs. We had a lot of patients who were like one patient with optic atrophy, one patient with sort of eye and brain defects. But the challenge, the real challenge was that in the UK, they don't do MRIs routinely on children with a developmental delay. But in the United States, we do it on everyone. So some of the kids had serial MRIs and some of them didn't. So there were some phenotypes that were actually a lot harder to assess because we didn't have sort of equivalent assessment of these different phenotypes. So the big question that this leads us to is sort of like, how do you think about variable expressivity or expansion of these phenotypes across in this cohort of patients or any cohort of patients? You can think about environment. We can think about the common genetic variation. I added this after this afternoon's talk. There's structural variations in STRs, and then also additional rare genetic variation. And all of these can change the way a phenotype is expressed in an individual. And that's really the level that we're trying to get to. What we'd like to get to is how can we think about, in an individual, can we predict or at least explain that phenotype based on the genetics that we have? So there are cases. There's an article in the New England Journal from about 4 years ago now that said of their patients at Baylor who had undergone clinical exome sequencing, they actually find that if a patient presents with what's an atypical presentation, so something where, okay, they look like they have X disease, but they have these extra features that have never been described before, that sometimes there's 2 or more rare mutations that contribute to the disease phenotype, which it's not that surprising, but we don't look for multiple rare mutations. There's a razor in medicine, Occam's razor, where you're trying to explain things with just one nice parsimonious explanation that explains the whole phenotype. And having multiple phenotypes, I think, makes things a little bit more complicated and harder to explain both to the patient but to the treating physician. So I hope I've kind of laid out for you some of the challenges around rare genetic disorders and how a lot of these phenotypes are morphing over time, and it's never clear if the phenotypes are due to the mutation that was originally described, or if these are sort of liabilities that are laid forth based on genetic background and environment and other factors that might be contributing to the phenotype that we don't routinely assay in clinical settings. So one of the things that we kind of embarked on when I started my lab was thinking about polygenic risk scores in rare disease patients. And sort of when I think about it, I'm like, I can't believe I thought this might work, because it was a challenging problem for many reasons, which I'll talk about. But the idea was that all phenotypes exist on a spectrum. So we think about height, and we know that people are short, people are taller. There's sort of a spectrum where the clinical phenotypes are at the extremes of short stature and overgrowth. And that's a very nice and clean example. But if you were to say, the example we were playing around with was congenital heart disease. And we were kind of curious as to whether or not we could develop a PRS for congenital heart disorders using some sort of related phenotypes, but not the exact congenital heart disease GWAS studies that exist today. And the question really becomes, like, how do you translate short stature and overgrowth? How do you define the edges of the spectrum? You might expect for congenital heart disease that you might have individuals who have abnormal valves to different degrees or abnormal structure, but at that everyone else on the more normal side, there's a lot of variation in cardiac structure that we don't assay, we don't measure. Some of it actually might be protective, and we don't really think about assaying it. So congenital heart defects, I'm going to do a little quick anatomy lesson. You have here, so the red and the blue are where your blood goes. There's some chambers in the heart. There's 4 chambers. They're connected with these valves that can be misformed. There's these big old arteries on the top, and if those are not in the right place or connect to the wrong chamber, you need pretty massive surgery to correct that. Sometimes the chambers don't separate properly, so you have one chamber at the bottom. But generally, there's a lot of different things that can go wrong. This structure derives from 2 different germline layers. And so you can have these if it works, it's great. It's kind of amazing that it works most of the time. You can have pretty mild abnormalities of congenital heart. So these are valve disorders, holes between the chambers that you can close up pretty easily in a non-invasive fashion. You can have your blood vessels that sort of supply your blood with heart. Those can connect in different ways, and it shouldn't matter. It usually doesn't matter at all unless you're having a procedure and they note it. That's when you might identify it. And then individuals might have thick or thin heart muscle, and those are all relatively mild. Sometimes they need clinical care, and sometimes it's just a watching and waiting. Sort of on the flip side, you can have more severe disorders where a chamber is missing, chambers are fused, the arteries connect in the wrong way. And then if anyone has any ideas of sort of protective mechanisms, something that makes your heart beat extra good, these are things that we haven't identified because I don't think we really have a good sense of what they would even look like. We don't assess sort of non-deleterious things in the clinical setting. But what I want to point out is that all these phenotypes exist on a spectrum. So what you can see here in there's a normal aortic valve, this thing that looks like split into 3 pieces. Normally your aortic valve looks like that. There's a handful of people, 1% to 2% of the population, that have a valve just has 2 valves. And this valve works totally fine but it has a higher propensity to undergo things like aortic stenosis and have what we call a heart murmur. You can actually hear it when someone is listening to your heart. And so these bicuspid valves usually will present themselves in individuals around 60 or 70 who need they have aortic stenosis and need an aortic surgery. So one famous example here is Arnold Schwarzenegger had a bicuspid aortic valve, so he's like a picture of health. But he did have this bicuspid valve that needed to be replaced, I believe, when he was like 59 or 60. And so these exist on a normal spectrum, and they're probably part of that, what we're calling normal in the general population. And so I want to make a point, too, that ICD codes are not good phenotypes at baseline. So when we're trying to look at GWAS that are ICD codes, that are based off of ICD codes, we really run into the issue in that most individuals who have what I would consider on the spectrum of congenital heart disease, they actually won't be coded as a congenital heart disease phenotype as an ICD code unless they have a really severe, like requiring surgery kind of phenotype, which is a really, really rare manifestation. The mild abnormalities like the bicuspid valve at birth, you won't see a clinical phenotype. It won't be caught unless you happen to be having an echocardiogram to look at the actual structure with ultrasound. So the question we asked was, can we use phenotypes that show the effect of congenital heart defects, these mild defects, without actually, we didn't want to use the congenital heart defect GWAS. It exists. There's like 300 or 400 patients. It's really unbalanced. It's not a good GWAS to use. And so what we ended up doing was we had two cohorts. We ended up using the UK Biobank summary statistics weights from two different, and I'll share with you exactly which ones we used. We looked at this cohort of cases of whole genome sequencing data for kids who had congenital heart disease and their parents. And then what we wanted to do was use these data sets to develop a polygenic risk score. So this is our subset of data. It was 711 trios, but we removed anyone who was admixed or non-European ancestry. And then because they were related, we ended up using pseudo-controls. So we have the proband, and then we'd use the untransmitted alleles to create these sort of fake people that were completely unrelated to the probands. And then what we did here was then to take this data, and we took a couple of different GWAS to see what we could do to build this PRS. And the idea here was that if you have a structural defect, even a mild one that's not fully penetrant, that maybe we could see it in abnormalities of heartbeat or sort of valvular murmurs, so milder phenotypes that might be encoded within the EHR. And we used the NeoLab versions of these GWASs, and then we also went into PheCode, some of the ones from the University of Michigan, which had a good number of cases to test this out. And we did our validation through permutation because we didn't really have a third data set. These are rare cases, so rather than split our data set again with our European data set, which is already pretty small, we ended up just doing our validation through permutation. So this are the GWAS, the results that we used. We have 3 of our PRS results that actually showed significance in our target cohort. And when we look at the distribution of PRS, you can see that the cases are actually a little bit lower in their PRS than the controls. And this was sort of a surprising result when we first looked at it. And we've been thinking a lot about it. I'm happy to entertain and think a little bit more about what we're looking at. And then when you map, so on the x-axis here, we have the decile of polygenic risk score. And at every decile, we looked at the odds ratio of having this congenital heart disease phenotype. And what you can see is that the lowest PRSs actually have the highest risk of polygenic risk, or of the heart valve phenotype of having disease. And our highest PRS was actually somewhat protected. That line, it doesn't actually cross 1. And so there's a couple explanations for this that could exist. One is that it could be a protective allelic effect, that we're not actually using a congenital heart phenotype. So maybe there's something protective about making a valve or a heart murmur in the way it's encoded. The other thought is we haven't taken into account, we removed any rare variants here. And so in the lowest decile, maybe everybody who has very little polygenic risk, we really should be looking for rare genetic mutations. And I should note here that the target cohort that we did use had already been filtered out for any known protein coding causes of congenital heart disease. So that cohort had been exomed. Anybody who had a diagnosis based on exome was then moved into genome, and that's the data set we used. And they published that data set in Nature Genetics in 2020 or 2021, and that data set was had a lot of they found maybe 1 or 2 non-coding variants in genes upstream of genes that are known to cause congenital heart disease, but it explains a tiny, tiny fraction of the cases. So the last thing we did was then to say, well, can we stratify our patients in our target cohort based on the severity of their disease? And so the pseudo controls were 0 because they ostensibly had no cardiac structural anomalies, and then we just designated whether or not someone was mild, moderate, or severe in disease. So you can see the 3 colors here. Green, blue, and purple is most severe. So it goes up a little, I don't know, it goes down a little bit in the same way. And our odds ratio follows that same trend, but it's not nearly as clean. And so this is sort of where we stopped this project at this point, sort of thinking about like how we can find better phenotypes or improve the phenotypes that we're looking at. And we've dabbled in a couple of other whole genome data sets with the same approach. And sometimes it works, and I think it really depends on sort of the quality of your GWAS data set. So that's sort of the last bit that I have here, but I kind of wanted to share with you because I was really excited by the James Webb Space Telescope images that came out this morning. So when you have these poor and incomplete phenotypes, it's kind of like looking at the Hubble telescope. It's a little blurry, it doesn't look great. When you have high resolution phenotyping, along with integration of maybe rare variants, you might get a much more beautiful vision of like the world in general. This was billions of dollars. I don't really have that much money to rephenotype everyone at that level, but one can dream. So the good news is there's still a lot of work to be done in germline genomics. I think we have a lot of there's a lot of work to be done both in the phenotyping space, but I think the other way you can look at this is to think about phenotyping at the molecular level in cell lines to try and build the case for what variants might be of interest. So future directions. I talked to you a lot about rare disease, which is really interesting to me, but we realize that rare diseases are kind of underpowered in this sense. And so one of the things that we're really looking at is sort of integrating monogenic variants with lipid traits and polygenic risk scores. And this is led by a graduate student, Angela Way, and co-mentored with Noah Zeitlin. So we've been doing this a little bit more in the UK Biobank, which is a really rich data set to kind of better understand how this might work on a rare disease level. And then we've thought about it, I mentioned incorporating additional rare disease whole genome sequencing cohorts. So the Gabriella Miller Foundation has a lot of other phenotypes out there, and they've been sequencing cohorts since 2015, different cohorts, different phenotypes, and they've expanded now to long-read sequencing and some RNA-seq, which might be of interest to anyone here. And then we have a wonderful big summer student right now who is looking into rare pathogenic variant identification in that low PRS cohort, trying to see if we can sort of think about, well, if you have a low PRS, maybe that's why, maybe there's a rare pathogenic variant that's explaining that weird signal we're seeing. So you guys are welcome to go to the beach. I thank my students, Sarah Spenlove, who did a lot of the work that I shared with you today. And just my lab for chugging through the last 4 or 5 years now.